All right, we're going to go ahead and get started. Once again, hello everyone, and thank you for joining us today. My name is Courtney Lindy, and I am the Digital Marketing Manager here at Alcami. I welcome you to today's webinar, Extractables and Leachables 101, the Past, Present, and Future. Today's session will be recorded and made available to you in the coming days. And if anyone has any questions along the way, please feel free to type them into the questions section of your toolbar, and we will be sure to answer those questions at the end. So at this time, I'm going to turn the floor over to my colleague, Cheryl Johnson, to begin today's presentation.
Thanks, Courtney. Hi, everyone. I'm Cheryl Johnson. I'm the Commercial Development Manager for Alcami's Biotechnology Services offerings. I support the large molecule offerings as well as the technical liaison for extractable leachables, and I'm excited to give everybody a crash course on all things extractable leachables today. And my computer froze. There we go. All right. The regulatory requirements for extractables and leachables have been evolving for decades. In the beginning, the scope of these studies was limited to the primary packaging because it's in direct contact with the drug product for extended periods of time. In-use componentry was also of interest as it contacts the formulation during administration. But the more we learn about potential sources of leachables and their potential for toxicity, the scope has broadened to include components used in manufacturing. In addition, with more and more biologics coming to market and the relative complexity of their manufacture, a comprehensive framework for determining the risk of drug product material interaction and systematic approach to addressing these materials with respect to potential leachables is warranted. In this webinar, I'm going to review the history of extractable leachables, discuss the best practices with current industry guidelines, and provide a brief overview of the developing guidelines. The very first question to answer is really simple. What are extractables and leachables? Extractables are compounds that can be extracted from a container closure system via laboratory manipulation, such as exposure to solvents or heat. Extractables are potential leachables. Leachables are compounds that passively migrate into the drug product over time as the result of drug product with the primary packaging, in-use componentry, or manufacturing surface. Migrants are compounds that may move from packaging components or materials into the drug product. They differ from leachables in that they accumulate in the drug product after crossing a physical barrier, such as the primary container closure or secondary packaging. An example of this would be the adhesive or inks used on a label migrating through a cyclic olefin vial into the drug product. The second question to discuss is whether leachables really put drug products at risk. In a word, yeah. There are many documented cases of leachable compounds interfering with drug product assays, diagnostic tests, and impurity profiles. Some compounds react with APIs, and others are known to cause harm to human health. While agency guidelines for new applicants are relatively well defined, we've also seen cases where a mystery peak showed up in the impurity profile of a commercial drug product, which was attributed to a leachable from primary packaging, or where leachables observed at the tail end of a stability program were traced back to the improper selection of a rubber stopper for a parental drug product. These are just a couple of examples to show why extractables and leachable studies are an important part of your application. Potential leachables can come from a variety of sources. Thus, it's helpful to understand the basics of how common primary packaging components are made. Almost all rubber used in the pharmaceutical industry today is synthetic, and the majority of closures are halobutyl rubber. It's made by combining raw rubber with cross-linking agents and a variety of additives to provide the desired properties. That's color, hardness, permeability, stuff like that. Any of these additives can find its way over time out of the polymer matrix and into your drug product. In addition, various degradation products are possible for each of those additives and cross-linking agents. While a manufacturer may know very well what goes into the rubber, it can be impossible to figure out how it will come out. The equipment used to manufacture drug products is also made using similar methods as the primary packaging. For example, the materials are exposed to mold release agents, colorants, cross-linking agents, and so on. On top of this, the components are often gamma irradiated or autoclaved prior to use, increasing the likelihood of compounds leaching into the formulation during manufacturing. As mentioned before, there are several well-documented incidents of contaminants leaching from containers and packaging dating back to the early 90s, and this led to the development of the E&L field. Next, I'm going to take a closer look at those landmark events and how the regulation has kept up. The first concerted effort to understand leachable compounds launched in the 1990s when the FDA became aware of elastomers used in metered dose inhalers leaching polycyclic aromatic hydrocarbons into the drug product. PAHs, if you're not familiar, are subject to regulation because they are linked to a variety of cancers. In 1999, the FDA issued a guidance for industry stating pharmaceutical manufacturers must show suitability of their container closure system for intended use, and materials must not leach harmful or undesirable amounts of substances to which a patient will be exposed. In 2002, it was discovered that diethylhexyl phthalate could leach out of the IV infusion lines into total parenteral nutrition. DEHP is a plasticizer and suspected endocrine disruptor that is used in the manufacture of PVC products, including bags and catheters for IV delivery, and transfer line and storage bags used in manufacturing. The FDA issued a public health notification restricting the use of DEHP after it was discovered that infants were sensitive to DEHP-triggered toxicity. 8 years later, Johnson & Johnson voluntarily recalled more than 500 lots of various over-the-counter products in response to customer complaints regarding a musty odor. The cause turned out to be the migration of a brominated fungicide in the wooden shipping pallets through the external packaging and container closure system into the drug products. This recall cost the manufacturer an estimated $900 million in lost sales. Working to come to a consensus on the approach to E&L is an ongoing process. The first official USP chapters directly addressing the design, justification, and execution of extractables and leachables assessments became effective in 2015 with chapters 1663 and 1664. Prior to this, the assessment of primary packaging with respect to leachables was limited to physiochemical properties such as non-volatile residue and extractable metals, as described in General Chapter 661, known as Plastic Packaging Systems and Their Materials of Construction. USP also introduced subchapters to 661, which delineate between materials of construction and packaging systems for pharmaceutical use. However, due to industry feedback, USP has since extended the implementation of this suite of chapters until 2020 and reinstated the original content of 661. An unintended consequence is confusion on which chapter to use for testing, which hopefully I'll clarify a little bit later in the webinar. The newest iteration of regulatory guidelines are USP Chapter 665 and 1665, which formally expand the scope of sources of potential leachables to manufacturing components and closes gaps in the preceding chapters. An overview of these chapters will be covered in the final section of this webinar, and a deep dive into the details of each will be covered as their content becomes final. It is ultimately the responsibility of the drug product application holder and drug product manufacturer to ensure the primary packaging and delivery systems are constructed of materials that are well characterized and suitable for their intended use. Therefore, it's important to understand the scope of each of these chapters and how it may apply to your application. Now I'm going to review that current guidance and some best practices for an extractable leachable study. Materials characterization and suitability for their intended use is a three-phased process. USP 661.1 covers the characterization of the materials of construction and their appropriateness for their application. The Extractables Assessment is first introduced in Chapter 661.2 with reference to 1663. And Leachables Assessment of drug products is covered in 661.2 as well with reference to Chapter 1664. Before going into these chapters in more detail, I'm just going to take a quick minute and review USP 661 because it's the chapter from which the remaining chapters are based. Initially, this chapter included specific test methods and specifications for the specific materials of construction and didn't differentiate between the material of construction and the final packaging. This created the distinct possibility of duplicate testing, and more importantly, it didn't establish a viable path forward for addressing extractables and leachables. When the USP introduced the subsequent chapters clarifying who is responsible for what, Chapter 661 was trimmed to include only a brief introduction and a scope for the applicant to use. Complicating matters, the USP reverted to the original content when it extended the implementation period until May of 2020. So depending on the phase and application status of your product, you may be able to justify using the original text of 661. However, with the implementation period closing less than a year away, early adoption of the subsequent chapters is the lowest risk to your approval timeline. USP 661 pertains to the plastic materials of construction only, such as the resin used to make a plastic bottle, and therefore extractables and leachables does not apply. This chapter addresses the ingredients used to make the packaging, not the packaging itself. The chapter provides test methods and specifications for specific materials such as cyclic olefins, PET, and PVC. Notably absent are cured polymers such as the silicone used for tubing and manufacturing. Each material of construction has requirements to establish the identity, bioreactivity, physiochemical properties, and composition. The final requirement includes extractable metals and testing for plastic additives, which are common analytes for extraction screens, but again, the scope of this chapter is limited to the material to make the packaging, not the final packaging. The scope of 661.2 includes that final packaging, and it's the base chapter for which extractables and leachables testing is derived. This chapter is used to establish whether the packaging system is chemically suited for its intended use with respect to patient safety. The chapter lists methods and specifications in 3 sections: bioreactivity and physiochemical testing, which are common with Chapter 661.1, and the third is the chemical safety assessment. The chemical safety assessment defines the expectations for testing the packaging system, but due to the diversity of drug products and packaging systems on the market, does not provide specifications. It does, however, provide the scaffolding on which the assessment must be assembled. The study design is expected to take the drug product formulation, dosage form, and packaging system into account when designing a study. The study must also include the appropriate analytical methods and be scientifically sound. The applicant is permitted to perform this testing on various configurations of the packaging system, such as the individual components, the system in entirety, or using the packaged drug product with scientific justification. Finally, it refers the applicant to Chapters 1663 and 1664 for extractables and leachables best practices. One important distinction is the requirements of 661.2 supersede those of 661.1. As stated in the beginning, that individual plastic materials of construction are deemed well characterized and appropriate for their use if they meet the requirements in this chapter or are used in a packaging system that meets the requirements in 661.2. And this is because the testing guidelines are more rigorous and tailored for specific packaging systems. Thus, if your packaging vendor is able to assure compliance with 661.1, only part of the work is done, and you will need to proceed to 661.2. But if for any reason 661.1 compliance is not specified by your vendor, you can perform the testing per 661.2 and kill 2 birds with 1 stone. The purpose of USP Chapter 1663 is to provide a framework for an extractables assessment. The guidelines are intended as best practices and do not address every drug product packaging configuration. The key characteristics of an extractables and leachables assessment yields a comprehensive analysis of the primary packaging system via controlled extraction studies that leverage current industry guidelines against the applicant's specific packaging configuration. The optimal extractables profile uses complementary analysis techniques to identify and estimate concentrations of potential leachables and will include a carefully selected analytical evaluation threshold, taking exposure limits and the dosage into account. Ultimately, the goal is to develop a worst-case profile of potential leachables for the specific drug product packaging configuration in advance of leachables being observed downstream. Because the goal of an extraction study is to build a worst-case profile, it's important to select conditions that will provide valuable information without causing degradation, because it can hinder meaningful analysis. The scope of the extraction plan should be tailored to the specific drug product based on the dosage, duration use, and the route of administration. This is because the risk for toxicity of a compound leaching from a metered dose inhaler is very different from that of a single-use parenteral. The classic approach to extractable studies involves exposing the componentry to a range of solvents under conditions designed to drive organic and inorganic potential leachables into solution for subsequent analysis. The solvents should be appropriately aggressive without damaging the base polymer. If possible, leachables analysis of aged drug product can be performed side by side to generate an extractable leachables correlation at the same time. This approach, while thorough and robust, is best applied to componentry that will be in direct contact with the formulation for the duration of the drug product's shelf life. When considering the transient exposure of the formulation and it may have during manufacturing, a simulation-based approach is more appropriate. A simulation study is the bridge between actively driving compounds into solution and the passive migration over time. A worst-case profile can be developed by simulating the conditions that extend slightly beyond the conditions a component will ever encounter during manufacturing. Ideally, one solvent is a drug product formulation or placebo, and the second can be an aggressive solvent to aid qualitative evaluation of the formulation sample. Simulation studies can also be applied to in-use componentry because the relatively short period of time the component will be exposed to the formulation. A full extraction, as described previously, in either case is not needed because the formulation will not be in direct contact with the component for extended periods of time. Regardless of whether the components of interest are the primary packaging, manufacturing surfaces, or in-use components, a single analytical technique is insufficient for the detection and quantitation of all extractables. It's important to use complementary analytical techniques to perform a truly thorough analysis. Chromatographic techniques coupled with mass spectrometry can yield powerful structural information for various classes of compounds. Each analytical method is designed to capture the broadest range of target compounds, and in many cases, the identity of a compound can be ascertained through the use of commercially available libraries, internal databases, and in-house expertise. In the case where a compound cannot be readily identified, basic structural analysis may be performed in order to glean any potentially useful information. In addition, the use of inductively coupled plasma mass spectrometry is an indispensable tool for the analysis of elemental leachables. Another critical characteristic of extractables and leachables is the analytical evaluation threshold. The AET is the single most impactful element of a thorough extraction study, as it is used to determine the appropriate sample preparation, analysis techniques, and most importantly, it serves as a threshold for qualification of potentially harmful compounds. The basis of the AET is a safety concern threshold published by various agencies that determine the acceptable daily intake of potentially toxic compounds. The most common SCT is used for genotoxic compounds, which is 1.5 micrograms per day per USP, PQRI, and ICH guidelines. The drug product specific AET is derived from the SCT, the maximum daily dose of the drug product, and an uncertainty factor to accommodate variations in the response of target compounds. This value is typically expressed in units per component or units per day. In the example below, where one vial is administered per day, the AET is determined to be 0.5 micrograms per day. This translates to the statement, we're going to take further steps to understand the risk posed by any compound if it is observed above 0.75 micrograms per day. This threshold correlates directly back to leachables, the final stop in the chemical safety assessment. The Leachables Assessment is the final phase of the assessment process. The goal is to evaluate packaged drug product that has been aged under shelf or simulated conditions for the same classes of compounds as an extraction study. While the extraction study builds the worst case profile, the Leachables Assessment focuses on the real life scenario. The Leachables Assessment is best performed using the same analytical techniques as the extraction study and can be performed concurrently if desired. Alternately, a more targeted approach can be designed based on the results of the extraction study. Either way, if an applicant is able to demonstrate that the drug product does not contain leachable compounds at levels of toxicological concern, then the Leachables assessment is complete. If compounds are observed at levels of concern, the applicant will have the benefit of foresight so that any steps needed to mitigate the risk can be implemented with minimal impact to their program. As investigators and agencies learn about potential leachables and their potential risks to human health, regulatory guidelines must be adapted to protect the public. It's important to keep in mind that future guidelines posed by USP Chapters 665 and 1665 are developing, but I'm going to cover them in brief next. As previously discussed, some of the gaps have been addressed in the 661 suite of chapters by splitting the testing of materials construction and the final packaging into two separate chapters. A comprehensive framework for the approach to E&L has been provided in these chapters, but some ambiguity around the manufacturing surfaces still remains. In addition, biologics manufacturing is highly complex compared to small molecule manufacturing and often uses many variations of single-use systems made of plastic. This alone warrants specific attention by the USP. The USP aims to answer some of these questions with draft chapters 665 and 1665, and they directly address the surfaces used in manufacture of drug products. The purpose of these chapters is to determine whether the components are suitable for their intended use, much like the chapters discussed earlier. The scope of Chapter 665 is applicable to all APIs, drug substances, and drug products, but it's limited to processes involving liquid. The chapter, in its current form, lays the framework for determining whether a leachable study is needed by directing the applicant to perform a two-phased assessment. The initial assessment is a paper study to determine whether the component has meaningful contact with the formulation and if the component is used to manufacture an approved drug product and marketed API, drug substance, or drug product. If the component is in contact with liquid during the process stream and its use in an approved drug product cannot be ascertained, then additional assessment is needed. Otherwise, the applicant should have enough information to justify eliminating the component from additional testing. The second round of the risk assessment is a more detailed review of the potential for interaction of the component with the liquid, and it defines the testing requirements based on the results of this assessment. The detailed framework for the assessment process and some additional guidelines for testing are provided in Chapter 1665. Draft Chapter 1665 is divided into material characterization, component characterization, and system characterization. Many of the characteristics of this assessment process is similar to that which is used to select the primary packaging by asking the following questions. Is the component made of well-characterized materials? Is the component at an elevated risk for leachables due to treatment prior to use in manufacture or exposure during manufacture? And finally, is the system in entirety qualified for its intended use with respect to process equipment-related leachables? The first two questions are simple enough to answer, and by referring to 661.1, 661.2, and 665, many materials of construction are addressed. But there's always the possibility of new materials entering the market, and USP provides some general information on how to handle these materials as well. The chapter contains an extensive process for evaluating the process train for potential leachables, and if a thorough safety assessment is deemed necessary, the testing guidelines are provided as well. The evolution of extractables and leachables regulation is a direct result of information gained by studying the packaging systems, in-use componentry, and components used to manufacture drug products. While a manufacturer may know what goes into these components, it can be impossible to predict what will come out. By performing a chemical safety assessment that is scientifically sound, employs appropriate preparation and analytical techniques for a range of compounds, and based on a sufficiently conservative AET for the drug product packaging system, the applicant can confidently determine whether the material is suitable for its intended use. With new USP chapters on the horizon, it's best to prepare for all the changes well in advance.
Great, thank you very much, Cheryl. We are going to begin the question and answer portion of today's presentation. And joining us for today's Q&A is Dr. Samuel Conway, the lead on our Extractables and Leachables team here at Alcami. So it looks like we had a few questions come in along the way, but feel free to continue to type them in. So the first question is, do the methods need to be validated?
The methods in their initial phases don't actually need to be validated. In the very beginning, the extraction studies are based on qualified methods that are intended for general use, and that's considered sufficient in the beginning. But generally, if you're looking at target compounds, say to monitor during stability, or even if you want to monitor for general leachables as a part of your stability program, then yes, specific methods should be developed and validated.
All right. And what are the requirements for ANDA submission?
Sam, you want to take this one?
They're pretty much the same for any submission. The regulatory agencies expect the drug manufacturer to have an understanding of the packaging, and sadly, they often stop at that description. If you're putting in an ANDA and you're trying to compare your particular drug to a reference listed drug, you're going to need to demonstrate that the leachables profile in your drug is going to be the same as that in the reference drug. So there really is no difference. In any phase that you're traveling forward, you still have to have that extractables and leachables information on your package.
Okay. And can you perform testing for 661.1?
Yes, we can. Because the chapter typically only applies to the materials, that would be performed by our compendial testing group. So yes.
All right. What is the best approach and/or recommendations for selection of extraction solvents? What aspects need to be considered and/or documented into the protocol?
Ooh, that's a good question. Well, we typically start by having an active dialogue where we discuss the specific formulation. We're looking for any key indicators such as high or low pH, if it's an aqueous formulation, if there's any solvents or surfactants and things like that. We pair that information with the daily dose and the duration of use. More often than not, 3 solvents can be employed. We typically like to include, say, 2 aqueous solvents that mimic the formulation and bracket the pH range that's acceptable for the final drug product. And then we use a 3rd solvent that is more aggressive to give you that worst case profile.
Okay. What types of materials does Alcami have experience with?
We've worked with a broad variety of materials. We've got experience with cyclic olefins, PVC, PET, polypropylene, silicone tubing, and of course the broad spectrum of drug products, parenterals, oral dose, nasal spray, and orally inhaled products, topical creams, ointments, you name it, we've got some experience with it. Sam, am I forgetting any?
My favorite, that would be elastomers.
Oh, of course.
All right. And how are your teams keeping up with regulatory changes in the industry?
We subscribe to a number of different industry journals, as well as keeping track on the USP Pharmacopoeia Forum and the ICH guidelines, and let's see what else. We stay in contact with all of the regulatory agencies as best we can and mine the publicly available information for any of those keywords for extractables and leachables.
Are there any published SCT values specific to topical dermatology products?
There's actually some ambiguity about topical products. As far as we know, the best approach is to use the most conservative available for parenterals, really, even though it's understood that the bioavailability of any compounds is significantly different. And you can use that as justification for a higher exposure level. Hopefully that's a good enough answer.
Okay, and the next question, are there guidelines for assessing leachables from stainless steel manufacturing vessels, and what is the current thinking on these materials?
Alcami's perspective is that the stainless materials, they're referred to as multi-use systems oftenly, and those generally fall under the category of elemental leachables, which we just essentially use the same techniques for that are referred to in USP Chapter 232 in the same way that you would do a risk assessment. And so what we typically do is there's a risk assessment process for the manufacturing, and we also analyze for inorganic compounds using the extraction design to pair those two pieces of information to say, okay, nothing is coming out that's inorganic from anywhere in the process stream.
Does Alcami have experience working on ENL for transdermal patches, and what are the challenges for the patch product?
We do. We have experience with them, and I think one of the single most challenging aspect of transdermal patches is the fact that oftentimes the patch itself is the drug product, and there are many layers to it because you have the active ingredient, you have any sort of adhesive, and then the base layer, and so we typically will do a study where we're looking at the material in entirety, mostly because the individual components aren't as easy to get a hold of as an analysis lab. So that's easily the most challenging part. And then coupled with the gray area around the regulation can make it a little bit difficult, but we certainly work through that on a case-by-case basis.
Are oral solid drug products subject to ENL screens?
Oh, this is a good question. So classically, many oral dose drug products have been able to get by deferring to 21 CFR's guidance on indirect food additives. However, with the implementation period closing for 661.2, the scope includes all drug products. So if you're a manufacturer or a developer on something that hasn't been approved yet, then you will be subject to it because it falls under the, those initial chapters of USP.
Okay, and how do you feel about the risks applied during cleaning and sterilizations of components and the risk of impact to the product potency, et cetera? And then second part to that question, do you feel the ENL study should be done with fully treated components, and if the cleaning/sterilization methods change, should a new study be done?
Yes, it's actually, so to start from the beginning, the cleaning and sterilization process is an unavoidable piece when you're setting up your manufacturing. And so our perspective is, and the agency's perspective is, that you must test the materials that have already gone through that process, because especially with the the high temperatures or the irradiation, it can actually change the surface of, say, your tubing or things like that ever so slightly. And so it's very important to perform your leachable study in a format that's already been through that. And I wouldn't be surprised if a leachable study was performed on materials that did not encounter those final stages, would be questioned by the agency. And so our perspective is, yes, it's important. And with respect to the potency and so on of the drug product, we have a team that specializes in material compatibility studies, and that's more focused on whether or not the tubing and the bags and so on have any effect on the drug product from that perspective, rather than having leachables come out of the tubing and affect it in that way.
Okay. Are there standard methods for determining the ability to measure contaminants in specific products, i.e., how do you know that your method is able to measure the contaminants in specific products?
Well, our current general screening methods are qualified for general use, and that essentially means that we've achieved the linearity and specificity and sensitivity for the specific methods in one type of matrix. If an applicant is looking at specific compounds, what we'll do is a spike recovery study side by side so that we're able to make that assertion. And typically the matrix that the samples are run in is the same as our standards and things like that. The question is, you have to close the gap between whatever preparation is required to get to that stage. We also use internal standards, particularly for our GCMS methods, to help ensure that you've got adequate recovery of any theoretical analytes. And this is actually a good question because a lot of this is based on the theoretical concentration of an analyte that may not actually be present. And so that's one of the reasons why it's really important to use a sufficiently conservative AET to help in those sort of differences in the matrix and the actual preparation procedure. And so when we design our studies, we take that into account to say, okay, we think we're going to get a theoretical concentration at this level, which is way below the actual level of concern.
Okay, and how the LOQ of the extractable method can be demonstrated without knowing the actual material of interest?
We use a broad range of commonly observed compounds. For example, our LCMS method for non-volatile compounds includes several phenolic antioxidants that we see fairly frequently. Our GCMS methods similarly use compounds that we see very frequently, and so we've qualified the methods based on those compounds. And part of the qualification process for the GCMS methods, for example, also included spiking some common matrices with compounds with a range of boiling points, so that we can say, we're able to see things across this range of boiling points, and therefore we've got data to support the I don't want to say the validity of the method, but the ability of the method to see these certain kinds of compounds.
Okay, and does the API manufacturer test all tubing, HDPE containers, inline filters used for in-process transfers during manufacture of APIs?
Typically, yes. It's important to note that with the new regulation around 661.2 and the subsequent chapters, that the scope of manufacturing analysis does include manufacturers of API. And so there is that risk assessment process. And the first question is whether or not it has meaningful contact with the componentry. And typically that's actually reserved to just liquid. And I believe there's also some specific assurances around drying off solvent, and there's some accommodations for that. But to answer shortly, yes. If it's got contact with all of the components, then all of them are subject to an extractable leachable screen. And that includes the tubing and the bags, transfer lines, the connectors used, filling nozzles, anything that has contact with the formulation during processing.
Okay, and does Alcami also have the capability to develop lyophilization cycles for drug formulation?
Yes, absolutely.
Okay. How long before NDA submission should E&L studies begin?
Well, typically, the earlier the better. We find that the best time is once you're entering phase 2. Once you've got your manufacturing train locked down, that's a great time to start thinking about that. As I mentioned before, we do perform material compatibility studies with a different team, and that's often a really good time because you're answering 2 questions. And then when it comes to primary packaging, once you've got your packaging system selected, there's little reason to delay, especially if you have a formulation that might have some solvents or solutions that have extracting power. And however, we're also capable of doing a comparison. So if you're trying to choose between 2 rubber stoppers, for example, we can certainly build a study that looks at both of them to be able to evaluate whether or not one might put you at a higher risk over another.
All right, and please clarify if AET is only applicable to leachable studies.
It's sort of a yes and no answer. I'm sorry, there's not a clear path. The AET is also the main reporting threshold, so you can base it off of anything as long as you've got scientific justification. And so, for example, with manufacturing studies, we often use a very, very, very conservative one, because taking the dosage and the number of vials or the volume of manufacturing the drug product into account, it ends up being a really, really large value. So we choose something more conservative based on those facts. And so the AET is looking at the end of the line, and so that's where the answer is yes, that it refers to leachable studies. And that's where, ultimately you're saying that my drug product isn't going to expose a person to compounds at above this particular level, but it's important to apply that threshold in the very beginning when you're talking about doing an extraction of your primary packaging. And so yes and no.
Okay. So a 2-part, can an extraction study be done using placebo material? And does agency accept leachable studies performed using a placebo material stored on stability?
That is largely dependent on the formulation of itself, or rather the active ingredient or the drug substance. We typically like to use the placebo. One of the questions that we like to answer early on, particularly with if you're dealing with a highly potent compound or a controlled substance, we consider that to be a good driver to use the placebo instead. So we look at the structure of the molecule that is your active or your drug substance to see whether or not it has the potential for leachables in and of itself, whether it's very low concentration or the structure itself doesn't have any indicators that it could potentially leach anything out of the drug product. We consider that if those answers are yes, that they do, then you need to include that in some capacity in your extraction study. But if you don't think that it's going to have any kind of interaction with the material, then you're justified to just do the placebo. And that is the same question for leachables. But we do strongly advise doing at least some level of analysis on the final drug product, because it's when it comes to leachables on stability.
Okay. Are manufacturing components of bulk drug substance typically included in Leachables assessment, or can these components be waived as part of a risk assessment only?
That is largely dependent on the drug substance itself. The most frequent requests that we work with are at the drug product manufacturing side, even though manufacturers of drug substance are subject to the same guidelines. So that would definitely depend on what the drug substance itself is.
Okay, and can Alcami perform a toxicology evaluation for leachables?
We currently do not do tox assessments in that way. We do make recommendations on what needs to be assessed, and we can refer you to a couple of different third parties that we deal with. It's our perspective that that can be better controlled if as a client you handle that side of things, just because of cost control measures and communication and things like that.
And do you need to use aged samples for the E&L study?
Yes, there's no harm in doing a quote-unquote time point zero or unaged drug product, but if you want to get a full view, we need to use aged leachables. The caveat to that is it doesn't have to actually be something that's at the end of shelf life. We can do accelerated aging in-house.
Great. These are all great questions. Do we have any more? What solvents should be considered for extraction studies? Are there any specific guidances on selection of solvents for extraction studies?
There are some best practices that are captured in 1663, but in general, the principles are that the solvents need to be adequately aggressive to mimic the formulation, or excuse me, bracket the formulation, and also provide that worst case profile. And so it's understood that specific solvents don't necessarily need to be included, but you need to have adequate scientific justification for the solvents that were selected.
All right, well, thank you all for your great questions. And if you have any more or think of anything later on, please feel free to reach out to Cheryl. Cheryl, if you want to head on to the next slide.
No, it doesn't want to. There we go.
Oops. All right. So you can see if you have any further questions, please feel free to reach out to our presenter Cheryl Johnson. Her email address is on your screen. That's cheryl.johnson@alcamynow.com. So I know a few of you had asked. The slides as well as a link to the recording of this webinar will be provided to you in the coming days, so be on the lookout for that. So with that, we'll say thank you all for taking the time to join us today and hope that you have a great rest of your day.